Boa tarde a todos.
Good afternoon, everyone. We are going to start the session on public health. I would like to thank the speakers and the audience. This session starts with Professor Mário Souza from the Multidisciplinary Biomedical Medicine. Good afternoon, everyone. Dear ladies and gentlemen, representatives of the Foundation for Science and Technology from the Parliamentary Commission of Communication and Science, National Agency of the Scientific and Technological Culture, the President of the meeting Science 22, the Ministry of Science and Higher Education from Egypt, our guests, and the colleagues that are the members of the panel and the public. This meeting, Science 22, is really relevant because we are celebrating the 25th anniversary of FTC, the National Scientist Day, and to honor our friend, Mr. Mariano Gago, who has been encouraging science of excellence in Portugal, with whom we were friends. So, on behalf of the Institute of Biomedical Sciences and Hospital of Porto, our greetings. Now allow me to introduce our research unit, a multidisciplinary biomedic unit which is headquartered in Porto, which together with the Porto Hospital Center pursues the goal of this meeting to establish bridges of knowledge between the Portuguese and the world, aiming love, humanism, ongoing improvement of well-being. Our motto is therefore to make a basic research for clinic and also a clinic research supported in lab. Therefore, it is a multidisciplinary area, as you can see in this image. We were funded by the energy of our dear Professor Nuno Grande, who has already passed away, a founder of our institution. We have two arms, one in the Institute of Biomedical Sciences and the other one in the hospital. The first one managed by Mrs. Monteiro, who is really a world-renowned person, and Mrs. Lobato, who is a neurologist, a very important figure in the world, namely regarding transplants and clinical assays or trials. So some of them, the transplants, were initiated in our hospital for the first time. During the last year, we have associated with two more units in order to have a new lab, associated lab, certified by FCT, ETA, which is managed by our renowned Professor Henrique Barros. In this second slide, you will have a real idea of the productivity of our organization in 2021. And I would like to underline not only the medical and scientific contributions with patents and so forth, but mainly the strong commitment that we have made to disclose science and to train and educate the youngest. This is the image that represents our planet. Advanced knowledge and permanent development of innovation is also the result of a world collaboration network between UNIB and other colleagues from almost all countries in the world in a global openness and without barriers. In this slide that you will be able to read carefully at home, which I will not speak in detail, you have a summary of their own main activities, research areas, and contacts for them to be able to get in touch with everyone. So, in our oncology group, we have our chairman of the regional northern unit. OMIB is proud to have several European reference centers, those connected with rare diseases, cancer, transplants, and family paramyositis. Regarding the latter, Professor Andrade, a neurologist in our hospital, was the person who discovered this disease, and then our colleagues were able to identify the mutagene and the protein, and then in Portugal was made the genetic diagnosis for this disease, and we obtained the first outcomes across the world. I will finish with a presentation of some examples of world premiere. At the top here, when we are able to discover the microorganism, and then how this technique became global everywhere, allowing treatment of the male infertility at a level never achieved, and then a collaboration with Spain. We were able, the first in vitro maturation of round spermatozoids, and after this date we began all the work with different groups across the world in order to one day be able to produce in vitro spermatozoid and eggs. Then we have a very funny image that has, we were living a time where the Dolly ship had appeared, and people were discussing if it was ethical to do it in humans. But we showed that technologically it was also possible to do in humans. Of course, this embryo is still frozen. Another aspect was that we were still discovering the relations of the chromosome with the male parameters, and with a group of a professor in Germany to discover the three main regions connected with the phenotype. And then with our group, with the Madeira Island, our dear Teresa, who has passed away unfortunately very young, we were able to find out a new region, the true responsible for most cases of the patients with this pathology. And I also have here another example, something new that changed the paradigm of science. Until then, we didn't know the origin of some diseases, and we demonstrate that in the germinative line, the first changes at the level of genomic imprinting take place, and then a series of diseases were explained. So you have had a perspective of the UNIB, and you are all invited to work with us. Thank you very much for your attention.
Vamos então passar à segunda apresentação. Ricardo Pinto.
Let's now move to the second presentation from Ricardo Pinto with his presentation. Thank you, Ricardo. He will talk about vinculation styles in adult age and the risk of infection by COVID-19, an exhibition of virtual reality. I will try to be as short as possible. Because we've been confined, I've been confined, I feel a little anxious. I think I'm not as confident as before. It might have changed the way we usually deal with presentations. Well, cortisol is definitely acting, but I feel a little anxious. Speaking of confinement, I bring a study today where we try to find some factors for explaining regarding exposure to COVID-19, as well as you can remember from the first lockdown, when we had the first cases in Portugal, we didn't need strict rules to make people follow the lockdown. People voluntarily chose to be in lockdown and to stay at home. We didn't expect it. We met an unknown reality. We weren't sure about the virus. So one of the factors that contributed for us to stay at home was fear, a perceived fear, a perception of risk that it would be harmful for our health. But after things got complicated, after several different lockdowns, because fear can't explain things alone, fear explains behaviors of protecting oneself, but literature tells us that messages that instigate fear are not that effective because there are other factors in explaining why people adopt healthy behaviors instead of risky behaviors. But there is an interesting thing. Vinculation has been used in psychology and psychopathology. But not as used in physical health. However, some studies, if we use this variable, have shown that people with insecure attachment and people with different traits are more involved in risky behaviors because they don't think over as much, so they tend to develop automatic responses. So people with insecure attachment and avoidant attachment are more involved in risky behaviors. One of the factors that explains this is the emotional relationship. So based on these premises, that attachment in adults, amongst adults, and an impulsive character explain the exposure to the risk of infection by COVID-19. So through an avatar, I will not explain it in detail now, but we had a pilot study. There was a study with several procedures, but the scenery was constructed to expose the person to a context. I'm not sure if I can make my avatar work, but there it is. It includes four scenarios. Normal context of someone leaving the house to go shopping. The person didn't have any information about being in a COVID project. Of course, at the end, there was that information, but initially is that you will participate in an experiment which involves doing everyday tasks. So just act as normally as possible. So the goal was to prepare dinner for friends, and on the way they would meet a friend, they would give this friend a lift. So everything the person went through the experience involved exposure to risk, being in contact with others, not disinfecting hands, standing in a queue. And let's remember that the avatar was tested during the peak of the pandemic. So the participants would have to choose between giving a lift to a friend or moving on, or would put on a mask. So while taking options, while making decisions, we would obtain a score, a score of risk. So whether the person chose not to disinfect hands. So this would contribute to a risk. We used several samples, including university students from the staff of university. So regarding the results,
what do you see? There is a COVID risk, what has been extracted from Avatar, and then we include these variables in a regression analysis. We realized that the fear to COVID is a variable that was significant, means the more fear against COVID or about COVID, less risk of behavior. And then we face an outcome, a result that made us think, did we do something wrong in programming the variables? But then we realized everything, we verified everything regarding the composition of the variables, and everything was okay, because the anxiety variable, the anxious link had a better positive, whilst the impulsiveness had a negative beta. This is strange. We don't understand why the connection, anxiety connection, the more connection or impulsiveness, the more impulsiveness, negative beta, less exposure to risk. So anxious link, the more exposure to risk, but impulsiveness was against what we had seen in literature. Impulsive people are involved in risk behaviors. So what did we do? We did a moderation between the fear of COVID and the total score of the exposure to risk. And here we understand already how negative beta, people with more impulsiveness, tend to involve less in risk behaviors. The more the fear increases, the most impulsive people, the red dots, tend to decrease the risk, whilst people with less impulsiveness tend to increase the risk. This result is a contradiction, making us think about the theories. What is the behavior of this variable? The theory that makes sense is the theory of the differential susceptibility. Based on the possibility of the differential susceptibility, more anxious people with anxious connection and more people more impulsive, they are more sensitive to the conditions of the context, of the environment where they are, for both good and bad things. People with difficult temper. We realize that people with difficult temper in a favorable context, they would benefit more from the situation, but on the opposite, in a prejudicial context, they were the ones who would be more harmed. People who are more impulsive, being aware that they are more impulsive, the more the fear increases, the more they have to self-regulate. They have to understand, I am a person who is very impulsive, so I will have to have some self-regulation. Probably this was what happened, but this is a possibility for future studies, and we will continue to study. But it's good to think that things are not linear, and in light of this theory, some people who think that they should follow psychopathology pathway, the context will invert the equation. Thank you.
Obrigada, Ricardo.
Thank you, Ricardo. I would now like to hand the floor to Constança Pombo. The title of the communication is Antibiotic Resistance in One Health, a Silent Pandemic that Threatens People, Animals and Environment. Thank you for the presentation. I will speak in Portuguese, and since we've had a previous session on this topic, in speaking Portuguese in a scientific context, I don't think anyone will be against. So I'm very pleased to be here and to talk about something that I've been concerned with for a long time. The COVID pandemic is one example of a One Health approach that affects people, animals, and environment. However, resistance to antibiotics is a growing problem with impacts on human health at hospital level, on the community, animals which are used for producing the meat we consume every day, on environment, but also in pets. What is One Health? The health of people, which is closely connected to the health of animals and our common environment. This approach is growing in importance, and I'll give you the example of a document that was recently published by the World Health Organization, whereby for the first time it became consensual that antibiotic resistance approach should be multifactorial in several areas and approaching human health, animal health, and environment. We know that there are two mechanisms that contribute to the emergence of antibiotic resistance: the use of antibiotic and the selection pressure, and the places where the most selection pressure happen contributes to the emergence of new resistance to new molecules in the studied populations. It is also very important, and this is a classical and very recent example, the resistance to colistin, which is considered a critical and very important antibiotic in human medicine. Colistin is used oftentimes more than what should be expected in treating infections for bacteria resistance to specific bacteria. This is a fact in Portugal, in Europe, in the world. This dissemination and this transmission between humans, between animals and environment has a specific paradigm in the colistin resistance, because in 2015 this resistance was related to the appearance of a gene, MCR1, a gene with a plasmidic origin. So this gene appeared in animals in China, in pets, disseminated in China for the first time in 2015. In 2022, this gene spreads rapidly around the world. It's very important to understand that this transmission has to do with the resistance mechanism. If the gene chooses to be in an element which can transfer across different bacterial species and across different hosts, then it will be successful. That's what happened with MCR1. This is a very representative diagram of the complexity of all the interactions, including the use of antibiotics at hospital level, at nursing homes, hospitals, primary healthcare too, the use and selection pressure in food production, in eating habits, in vegetable foods and environment. We have been focusing on recently, because we believe that this dynamic and this proximity, this familiar proximity of the presence of pets in our homes that have their humans and that look after us was one of the reasons, one of the targets reasons for this consortium we recently ended in October 2021 about the risk of transmission to antibiotics in pets in proximity with their people in a domestic environment, a domestic family environment. We reached the conclusion in this study, an international study, with the participation of several colleagues in the UK, Canada, etc. We understood that there was a low level of antibiotic resistance, and we're talking about resistance to antibiotics that we desperately need for hospital use in human medicine that sometimes are not available for veterinary use. So in these molecules, which are not the first choice but are the first line in the treatment in infections treated by multi-resistant bacteria, and we've seen that this transmission was low. However, this low transmission is a concern because we've been able to demonstrate that pets are a reservoir of the presence of these animals that might be transmitted to animals. Our lab, from this project, Pets Risk, we decided to focus more closely on the transmission and the transmission of antibiotic resistance, specifically colistin, and study it in the context of One Health. This context involves to carry out the monitoring of resistant bacteria in humans, pets, and in the environment, hospital environments, for example, common to humans and pets, and for individuals in the community. Also, the environment, including all this dynamic. The lab has been developing work in this area, and our vision is to try to take the genomics to prevent carbapenem colicin resistance to transmission and identify these links of transmission in the community and understand which setting, which population, which ecosystem is the most complicated and problematic in terms of amplifying the resistance. As recent outputs, I'd like to report the description of the MCR1 gene, which is vehiculated in this. In pink, the plasmodium detected in a bacteria of animal origin, and in blue with human origin. What did we detect? The bacterian genome was exactly the same in 2 transmission cases, and on the other hand, the plasmodiums, where the MCR1 were also the same. This transmission happened across people in the same house and across an animal which lived in the same house. Lately, we've also had the work of Joana Moreira da Silva, also reflected the importance of our work in microbiology in veterinary to know which are the resistant bacteria and to study its susceptibility to antibiotics. This is important because the first detections were in bacteria which infect pets, as KPC3, which was found. It has a genetic code similar to those found in Portuguese hospital, and Plasmodium is not exactly the same as the Portuguese Plasmodium, because what's in the database was the one isolated in a French hospital. So it's very interesting and also scary that there are serious consequences for this sharing, and that there is a mobility in bacteria and genes across two populations, such as pets and their humans. What can we do, and what is our goal regarding our research? We need to shift from a research which allow a complex bioeconomic analysis, and to be able to transmit that information to veterinary medicine in the context of One Health, and to be able to prevent the transmission of those multi-resistant bacteria. Obviously, we need to restrict use and preserve antibiotics, and be cautious about using antibiotics. So that's our three main areas: humans, animals, and environments remain healthy. And before ending, I would like to thank everyone, all the investigators that collaborated with us in Pets Risk, and that collaborate with us nowadays in our research strands. And I would also like to be very thankful to the pets and to their families who collaborated in our study. Our four-legged friends, it's difficult to explain that they have perfectly healthy animals, but they might have a multi-resistant bacteria. It's difficult to explain in terms of communication. There is no danger in having a pet. So the first thing they ask is, what should I do? One thing that happened during this pet risk, during these last years of research, we managed to identify actions and measures, basic measures of hygiene and infection control that we might institute in our house in order to prevent this infection. Thank you for your attention.
Obrigada, Constança.
Thank you, Constança. Now I will pass on the floor to the speaker from Gulbenkian Foundation. Thank you for the invitation, for the introduction. I'm going to speak in English because it's easier for everyone, I think.
I changed the title, by the way. I choose to present here an example of what we have done over the last 2 years. The Institute Gulbenkian Sciences, like many institutes in health, have invested in helping society facing the COVID pandemic. And one of our flag projects is this InfoVac, where we actually follow 3,000 individuals since the beginning of vaccination in Portugal. Follow means we monitor them at the beginning, before the vaccination, at every single dose, and at specific time point. I will develop this. And we have several questions to ask there. Some are a bit fundamental, because I am an immunologist and I still have curiosities upstream, I don't know, of societal health. But some are relevant for the management of the crisis. Some are hopefully relevant for the management of other vaccination campaigns. So I really just will show some extracts of what we do. Here it's I represent the two cohorts that I'm going to discuss today. One is a cohort of healthcare workers at the Schlo hospital in the Lisbon area. and the other one is a collection of nursing home residents. And as you see, we have a timeline since the beginning of vaccination, early in 2021 or even in December 2020 for some healthcare workers. And the diamonds represent the vaccination and sample collections, the circles sample collections. And as a visual first approach to that, this is the curves at the bottom of the slides are the dynamic of the antibody response to vaccination. We can see the shaded area each 1 month after a given dose of vaccine. We are following here, concentrating on these slides to the Pfizer vaccine that here in Portugal was given a regimen of 2 doses at 3 weeks intervals. And then time passed. The 3rd dose was given by the end of 2021, let's say. And what we see here, the last time point is 1 month post the 3rd dose. So you can see really the wave going up, going down. You can notice the stars. The stats here indicate that each dose brings the antibody level higher. We can see that there's a spread, so we are here on the log scale of the IgG titer. So there's a spread at each time points, which means there's heterogeneity in the amplitude of the immune response. And therefore there's many questions we can ask that could be relevant for the management of vaccination campaigns to this case, or to any follow-up if it will finally be reduced to more personalized approach, for instance. So a first question to understand the heterogeneity is to ask whether there is a sex effect, of course. It's relevant because immune responses in many contexts are stronger in females than in males. This is, for instance, one of the reasons why autoimmune diseases are more frequent in females than males. However, with this vaccine, there is actually no differences in the level of immune response induced in male and females. I'm not showing you all the details of the analysis and the stats, but this was a surprise, and good, probably. However, if we now ask the age effect, and because we had 2 cohorts, one healthcare workers, other nursing home residents, what we can see is that at any time point we measured, there is a clear age effect in the sense that aged people have a lower immune response to the vaccine, which is a good vaccine. We can ask what are anything in those profiles that could serve as an index of predictability. In this case, I'm illustrating the predictability of the decline phase. So the decline phase is what we see here between the response to the 2nd dose and before the 3rd dose. We have 4 time points, so it's pretty good to study slopes or more complex shapes. And the visual effect of what is represented in multiple colors here is to say that there's a quite regular decline in the antibody level. So as much as we have heterogeneity in the level of the response, we don't have much heterogeneity in the declines, as if the process is conserved across age, it's also across sex for what we tested. And this is interesting and useful because it makes that we can probably make predictions out of very little information. Predictions on the decline, predictions, actually we checked this also on the amplitude of the response upon a boost. Here the bottom scheme illustrates that with these strategies already very simple here, and we are working on more sophisticated approach. we can predict for a person when it will reach a threshold of negativity for its antibody. Here we say threshold of negativity. It was defined out of testing 1,000 samples, sera samples, pre-pandemic, so 2015 collection. And that's how we define this level is never found pre-pandemic, therefore above it's positive. And that's what makes us optimistic that eventually we can develop an algorithm that will be personalized to design vaccination protocol or regimen, what is the time lapse between 2 injections, for instance, that will be adjusted as well to the variants of concern circulating. So that's the type of work we are doing. There's much more I could say, but I think for the principle of the purpose of this presentation, it should be enough. And I will just acknowledge member of the team at the IGC on the left. Very tight collaboration with Telmo Nunes, who actually is the one who made possible the collection of nursing homes. João Faroviana at the Schlo Hospitals was the one who motivated all the healthcare workers to contribute to the study. Paola Alves at the IBET, who is the one that made possible for us to conduct these antibody measurements. We had set the ELISA ourselves with our academic tools, and IBET is specialized in producing very high quality protein and other molecular biological compounds, and produced the spike for us. This ELISA has been made available to the academia as well as to Medinfa as a company, and we at IGC transform it for very large scale analysis like this longitudinal large cohort requires. Then of course all the donors, participants to the studies, and the nursing home staff that help us for this. The bottom logos are one way or another. Those logos are institutions that help us do this work through funding, and that's where I will stop.
Obrigada. Passamos agora à Anabela Borges, voltamos à multiresistência.
Let's now move to Anabela Borges. Let's go back to multi-resistance. She is from the Lab of Biotechnology and Energy, and this is a nature-inspired approach to face the paradigm of multi-drug resistant bacteria.
To share a little bit about our research line. Today I will talk about a nature-inspired approach to face the paradigm of multi-drug resistant bacteria. Briefly, only to contextualize, as it was highlighted by Professora Constança Pomba, the problematic of antibiotic resistance attains now a global scale, which makes very difficult the treatment of a wide range of infectious diseases. In addition, this problematic is more evident in the current pandemic times, particularly because the overuse of antibiotics increase, which impose selective pressure on bacteria and also exacerbating the multi-drug resistance and pan-drug resistance occurrence. So this is a global problem that requires a global solution. However, although of the several actions and initiatives that have taken place in the last decades worldwide, it becomes to be not enough. And this is particularly difficult when you talk about special microbial communities that is known as biofilms. So in a simple way, a biofilm can be defined as a complex and structured microorganisms that could be adhered to a substratum, to an interface, or to each other, or involved in the matrix of polymeric substances produced by the cells. This process occurs in several sequential steps and is a dynamic and multicellular process. At clinical
Por
level,
exemplo, a nível
biofilms can
clínica,
occur
vemos que
on
os
the
biofilmes
surfaces,
podem
secretions
ocorrer
or tissues,
em
like
secreções,
wounds,
superfícies ou tecido
dental plaque,
localizado, por
or
exemplo, placa
medical
dentária
implants.
ou implantes
They are
médicos.
responsible for
São
more than 80% of
responsáveis
human infections
por
and
mais de
is
80% das
considered
infecções
the major
humanas
cause of
e são
persistence
consideradas a
and the
principal
recurrence
causa
or
de
relapse of
persistência
infections,
e de
leading to
recorrência
infections
clínica de
chronicity
infecções,
that share a
levando
common
à
characteristic of
cronicidade
tolerance
de
and
infecções
recalcitrance
que
to
mostram uma
antimicrobials
tolerância
and
e
also to the host
resistência
immune system
e
defenses,
também
which is
ao
known as
sistema
microbial biofilm
imunitário
recalcitrance.
do hospedeiro. Este fenómeno chama-se recalcitrância.
Regarding biofilm recalcitrance to antimicrobials, there are a lot of mechanisms that can be divided in tolerance or resistance. Tolerance is related with a transitory effect that is mainly imposed by the metabolic heterogeneity generated within the cellular population, impaired mutation of antimicrobials, and also due to an adaptive response. In another way, resistance is not transitory and is imposed mostly for mutations and horizontal gene transfers. Considering that these biofilm infections, recalcitrance, have a multifactorial effect, the process of drug discovery has been faced with a lot of
tem sido
difficulties.
muito difícil.
So, in this way, you need new strategies, particularly more innovative and differential. In this context, you can use natural compounds from plants due to their particularly chemical diversity and also differential mode of actions and also their ability to target multiple targets on the cells at the same time, which can provide several advantages compared to the chemical compounds.
Vantagens em comparação com os químicos.
Our group already demonstrated that phytochemicals can have multi-targets on biofilms, interfering with the several processes of biofilm formation, differentiations, and so this could be a good strategy. But today I will focus only on the inhibition of a mechanism that is related with bacterial communication, because this mechanism is interrelated with almost all the others.
Com quase todos os outros.
So, in this study, we tested furbina, that is a natural compound obtained from sugarcane bagasse, and this compound demonstrated good ability to interfere with the bacterial communication of the pathogen Pseudomonas aeruginosa, and this effect is obtained at the subinhibitory concentration, which is very relevant. It was also verified that by in silico studies that furbina competes directly with the auto-inducers, so this effect can be due to a competition with the auto-inducers produced by the Pseudomonas aeruginosa.
Desta forma.
It was also observed that this fluorescence inhibition effect could be correlated with an inhibition of the initial adhesion of bacteria and also with a decrease on biofilm productivity, and it could increase also the susceptibility of Pseudomonas aeruginosa biofilms to antibiotic ciprofloxacin. It was also observed that furbina inhibits the production of a virulence factor that is coron sensory related, that is pyocyanin. So this molecule is able to interfere with the ability of Pseudomonas aeruginosa pathogen to infect the host and also to form these complex structured communities that is biofilms. In another study, the same molecule and 4 synthetic derivatives was tested against another coron sensing system,
outro
from
sistema
Staphylococcus aureus and show also good results in both in vitro and in silico studies.
em estudos in vitro e em sílico.
This molecule was also able to decrease the biofilm formation propensity and also affect the susceptibility of the Staphylococcus aureus biofilms to a topical antibiotic that is succeedic acid. And thank you for all entities and
Muito obrigada às
collaborators
entidades
and
que
for your
connosco colaboraram
attention.
e obrigada também pela vossa atenção.
Obrigada, Anabela. Passo agora ao Miguel Salles Dias, do Centro de Investigação em Ciências da Informação, Tecnologias e Arquitetura do ISCTE, com aplicações móveis baseadas em inteligência artificial para respostas de saúde pública.
Muito obrigado.
Thank you.
Vou começar por dizer que é um prazer estar
It's
aqui
a pleasure to be
saudar,
here to greet
as pessoas presentes
the
e os caros
people
colegas
attending
do
the
painel.
meeting and my
Este
colleagues
projeto é
at
um
the
projeto
panel. This
inspirado
project is
pela
a project
situação
inspired by the
pandémica,
pandemic situation that everyone knows.
Começa mais ou menos há um ano.
It started
em
1 year
início de
ago,
2021.
beginning of
Chamámos de
2021,
aplicações móveis baseadas em
mobile
inteligência
applications
artificial
based on
para resposta de saúde
artificial
pública.
intelligence to respond to public health.
Ora bem, ele reúne uma parceria
It gathers a partnership, multidisciplinary
porque
partnership,
nós enfrentamos
because we are facing some problems and we also present some solutions that require holistic approaches. We have at ISCTE a group of computing, engineering and social area. We work with the Telecommunications Institute of the Instituto Superior Técnico, which brings a component of procedure of signal and sensorization, and also with colleagues of the clinical area or field with the Faculty of Medicine, particularly those who work in the cardiology unit at Santa Maria Hospital. We are also associated with the International Association of Artificial Intelligence. This partnership has been useful to address what I'm going to show you.
Portanto, o problema que enfrentamos aqui neste projeto, podemos enunciá-lo com estas duas perguntas de investigação. Portanto, a primeira,
the first one, how can we identify
pacientes
symptomatic or asymptomatic patients from biometric data, physiological data gathered from the citizens, to help a diagnostic type of medicine, but also a second question oriented to a prognosis. How can we forecast situations of decompensation of certain syndromes, particularly in this case, to prevent situations of infections by COVID, to advise in advance, even days, the citizen and ask him to make a test. So we focus these research questions in two types of scenarios. For now, COVID patients which are in remote treatment at home, or risk groups which are not infected, namely diabetic people, hypertensive patients, or patients with heart disease. There is a need to understand that when the project started and we launched these questions, we were already ending the lockdowns. We were still with the protection measures and social distancing, and society was already going back to their activities. And we had already distributed
por todo o mundo
across the world
aplications
applications
de
to
detecção
detect
de
contacts,
aplicações essas
which
que tiveram algum
applications
sucesso
which
interessante
were
em certas culturas
successful
e
in
certos
certain
sistemas
cultures
políticos,
and
em particular
political
na Ásia,
systems,
mas que não tiveram
particularly Asia,
sucesso
but were
algum
not successful
conhecido nas democracias ocidentais
in
e na
Western
cultura mais
democracies
ocidental,
or in
incluindo
the
também
Western
dos Estados Unidos
cultures,
e noutras regiões do mundo.
including the United States and
E, portanto,
other globe
o
regions.
nosso interesse
So our
seria,
interest
se
would
calhar, não
be
abordar
maybe not
este
to
tipo de problemas
approach
da mesma
this
forma.
type of problems in the
Portanto,
same
apesar
way.
de termos
So
aqui grupos de
despite we
engenharia,
have engineering
também
groups,
nos preocuparmos em olharmos
the concern was
para as
also to
questões da
address
aplicação pela
issues
sociedade
of
de certos tipos de
acceptance of
e também
certain
sempre
solutions by the
ligado às
society
questões clínicas.
connected with clinical
Muito bem, então este é o problema.
issues.
Então
So
como é que
this
nós
is
o
the problem.
endereçamos?
How did
Endereçamos,
we
obviamente, com uma
address
artilharia,
the problem?
digamos, com alguma
We used
complexidade
the complex
de
system,
sistema de engenharia, não é?
an engineering
que
system
tem elementos
that has
baseados
elements
no
based
smartphone. Portanto,
in a
no fundo, nós
smartphone.
vamos,
So we
estamos a
are
desenvolver uma app
developing an
e
app.
perguntarão, está bem,
an
então,
app.
e não tendo tido as outras apps de sucesso, o que é que
And
poderia
why
levar esta app a ter
should this app be accepted
precisamente
and not the
esta
other ones?
arquitetura é uma
This
arquitetura que
architecture
tem grande
is an
esforço na segurança de informação,
that
na
made
garantia
an
da
effort
privacidade dos dados.
in
Portanto, é uma
security
arquitetura que
information and
consegue
securing information.
claramente
We
aderir a
clearly
regulamentações como o Regulamento Geral de Proteção de Dados.
comply with
E
regulations in
baseada
terms of data
em
protection
tecnologias
based on
que são as que se
technologies
conhecem, que
that are
garantem
the ones
essas
we
prerrogativas, como tecnologias
know,
que
and
permitem
they
fazer a
really
rastreabilidade
we
e
can
auditabilidade
track
das transações usando
transactions
blockchain.
using blockchain,
E é claro
and clearly
que recolhendo dados
collecting
fisiológicos através do
physiological
smartphone ou
data
sensores
through
emparelhados,
smartphones,
depois usa
then
tecnologias
the
de
use
inteligência
of
artificial
artificial
para poder
intelligence and
fazer
technologies or technology
aquela
to
resposta
do
que eu
or to
referi,
give
que
the
é
answer I
às
said
questões de
before
melhorar o
in
diagnóstico,
order to improve
quer à
diagnosis
previsão
and
de
to also
descompensação
forecast
da
the
situação
compensation
clínica
of the
a
clinical
partir da
situation
análise dos dados.
from a data
E,
analysis.
portanto, é uma
And
arquitetura
therefore,
de
this
referência que nós estamos a trabalhar,
architecture is
que estamos a
quite
propor em sede também
important because we are
do
proposing
ENPI, que é
to
a
AMPI,
Associação Internacional
the
de
International
Padronização, está a ser muito
Association
bem
of
aceito.
Standardization that is being
Gostava
accepted.
de
And I would
mostrar
like to show
alguns
you
resultados
some of the
preliminares.
preliminary
Nós,
outcomes or
claro
results.
que não somos os
Clearly,
únicos
we are not the only ones
a ter este tipo de
having
abordagem.
this type of
Quando nós
approach.
começámos,
When we
outros
started,
grupos
other
internacionais também
international
fizeram
groups
o
did the
mesmo
same.
género de
The
abordagem.
similar
Por
approach.
exemplo,
For instance, Stanford
lançou
launched
dataset
a
público anonimizado
public data set
com 2.000
with
participantes
2,000
que recolheram dados
participants. They
de
collected
dispositivos
data
biométricos,
from
como
biometric
telefones,
devices such
portanto,
as
relógios e outros
watches
dispositivos
and
que permitem
other
recolher dados em tempo real,
devices that
dos quais
allow
84
us to
participantes
collect
com
data
Covid
in real time.
e recolheram o
And
batimento
they
cardíaco,
collected
o movimento,
heartbeat
só pela análise
movement,
do
and
batimento
only
cardíaco,
by
conseguiram, nós replicámos essa
heartbeat, and we
experiência,
replicated the
conseguiram
experience,
58%
they
de
achieved
previsão de
58%
verdadeiros positivos
in being able to forecast.
para passarem de uma situação sem Covid para uma situação com Covid. Como podemos ver ali
So as you
neste
can see,
gráfico, na zona
we were
verde,
able to see
temos
when an individual
indicador que
can
mede
suddenly
a
be
situação
infected
de
with
batimento
COVID
cardíaco em repouso, portanto, uma situação
by
normal.
analyzing the
E o nosso
heartbeat.
indicador
Our indicator
consegue,
is
estão a ver
able,
quando
as you
estamos na
can
zona
see,
vermelha,
we are in the
é
red
quando
zone.
o paciente está com
When the
Covid, o
patient
nosso indicador tem
has
pico.
COVID,
E com uma melhoria
our
de
device
técnicas
has a
de
peak.
inteligência artificial,
With
nós já
using
atingimos à volta de
artificial intelligence,
80%
we reach
de
80%
verdadeiros
of
positivos.
true
E
positives.
pensamos ainda
And we
melhorar
think
pouco
to
mais
even improve
este
this
resultado,
even further,
utilizando também
using
outras técnicas.
and other techniques.
Estamos também
We
a
are
fazer
also
em virtude destes modelos de
based
previsão
on
serem
these
pessoais, portanto, são
models,
baseados
because
nos
these
dados
forecast
do próprio
models
cidadão,
use the
e a
data
nossa
of the
arquitetura depois
citizen themselves, and
garante,
our
até mediante
architecture
contratos com
ensure
tecnologias de
by
blockchain,
signing
que o cidadão
contracts
possa
with
ou
blockchain
não
technologies
dar
for
autorização que esses
the
dados possam ser
citizen,
usados
giving permission
pelos
that
cuidadores formais
the
e informais,
data will
para
be
além
able
do
to be
próprio.
used
Estamos a
by
usar
those
uma computação no
blockchains.
próprio telefone.
So we are
Portanto,
using
há
an app
tendência,
in the
até
telephone itself.
pela capacidade que os
For the
telefones,
capacity
smartphones dispõem de
smartphones
computação,
have
de podermos
for us
distribuir
to be able to
o cálculo
distribute
destes
the
modelos
calculation
de
of
previsão
these
para o
forecasting
próprio
models
cálculo
in order to
smartphone,
calculate
sem
in
prejuízo
the
de existir
smartphone
também uma
without
arquitetura na
prejudice
nuvem que
of
possa
of
complementar.
existing an architecture in the cloud
E,
that can
portanto, nós
complement.
neste
So,
momento estamos a acompanhar o
we
estado
are following
até
up
estamos
the state of the
iremos
art,
publicar
and we will
proximamente,
be
estamos a
publishing
avançar pouco
because we
mais.
are advancing even more.
E também estamos a fazer
So we
recolha de
are
dados,
collecting
usamos estes
data.
dados de Stanford,
If
mas também já
we
recolhemos
use
dados no Hospital de Santa Maria,
We have
depois
collected data
de serem
in
aprovados,
Santa
obviamente,
Maria
pela Comissão de
Hospital
Ética.
after being
Temos
approved
já
by the Ethic
disponibilizado
Commission. We
de
have
aceder a 62
already
GB de
permission to
dados
have access
de
to
pacientes,
many data of
diversos dados em
patients
tempo
in
real, como a
several
temperatura,
areas,
batimento
even
cardíaco,
the
o nível de
temperature, the
oxigênio do
heartbeat,
sangue.
the
São
level of
dados que nós
oxygen
vamos
in their blood,
processar
data that we are
com as
going to
técnicas de
process
inteligência artificial
with the
para criar a
techniques
maioria
of
dos nossos modelos
artificial intelligence for us to be able to have our diagnosis devices and
Estes dados
prognostic
foram
devices.
recolhidos,
These
parte
data
deles,
were
na
collected
unidade de cuidados
from
intensivos
the intensive
de
care
Santa Maria,
units in Santa
durante
Maria
o período de Covid, portanto, como vêem ali,
during the
no
COVID
período de
period
2019
from
até
2019 until 2021.
Mas a questão que
The
levou
issue that
à falta de adoção deste tipo de soluções pelos cidadãos
the
não se
issue
prendeu
the
apenas
citizens
com as
didn't
capacidades
approve
tecnológicas
this only
e
because
as
they
funcionalidades,
didn't
digamos
believe in
assim,
the
da
technology.
solução. Prendeu-se também com
The reason was
o tipo de mensagem
the type of
que
message, the type of message
a própria
that
aplicação
the
transmite.
app
E, portanto, nós temos
conveys.
feito
So
estudos
we have been
e chegámos
doing
à conclusão,
studies and we
portanto, sobre a adoção,
concluded
estudos sobre a adoção
studies about
deste
the
género de
adoption
aplicações
of this type of apps
pelos cidadãos em
by
geral.
the citizens in
Chegamos à
general.
conclusão
So we concluded that
que as
the
mensagens
messages
que reforçam
that
a
reinforce
segurança dos dados
security of
e a
data
privacidade
and privacy,
portanto, tudo que a aplicação possa informar
everything
em termos
that the app
quer
can
diagnóstico,
inform,
quer
both in
prognóstico,
terms of
funciona
diagnosis and
muito bem
prognosis, they
em
work very well
populações que têm
in
risco,
population
são
that
exatamente
are
o
at
segundo
risk.
grupo que o nosso projeto endereça.
The sec-
Populações que estão em risco,
population
os tais
are
hipertensos,
at
os tais
risk.
diabéticos,
The people with
outras situações de
diabetes
risco,
or with high
elas
blood pressure.
têm previsivelmente, e nós já chegámos à conclusão que é estatisticamente significativo, que este efeito
significant
bem
number of people.
à adoção destas aplicações
They adhere very
quando
well to the
mensagens
adoption of these apps
melhor de privacidade e
because
de proteção dos dados são
if they can have
reforçadas.
their data
Portanto,
secured,
que nós
they are more
queremos seguir.
willing to accept
E então
these
o
apps.
projeto
That's
ainda não
why we
terminou.
have included
Vamos ter
this
mais,
security
possivelmente, talvez mais um ano, em
measures.
princípio,
So,
e
in a
esperamos
year,
ter
we
resultados
hope
mais
that
interessantes
we
e que a nossa
will be
aplicação
able to have
tenha
more
uma adesão
interesting
também
results, and we
interessante.
hope that our app will be
E muito obrigado pela vossa atenção.
accepted as a whole. Thank you very much for your attention.
Vamos passar então à última comunicação.
Let's now
Alessandro
move
Fantoni
to
e
our
Arnaldo
last
Batista, do
speech
Centro de Tecnologias e Sistemas,
from the
Sistemas
Center of
Ciberfísicos para
Technologies and
Monitorização em Sistemas de
Physic
Saúde.
Systems. Thank
Sim,
you.
muito obrigado.
Thank
E
you.
pronto, irei. Boa tarde a todos. Pronto, vou aqui apresentar.
Good afternoon, everybody. I will present some of the results obtained in our research center about cyber-physical systems for healthcare monitoring. This is the main topic, main focus of our research center. I will present the results of our monitoring. Our point of view is that these cyber-physical systems might represent an opportunity to try to comply the UN goals for 2030 in health, as they might be a useful means to try to overcome limitations related to geographic conditions or difficulties in accessing healthcare. So this is a set of new technologies. There is a hardware component. There is also a software component, an algorithm, transmission of information, and remote computing. The two cases I will present here are related to disease within nephrology, acute kidney insufficiency, and the other on pregnancy monitoring in women. Regarding acute kidney injury, the project aimed at developing sensors able to detect with biosensors, both based on technologies, plasmodic, the first with particles made of gold, and the other based on different circuits. In both cases, the innovation that we present is especially in the way the information is extracted from the sensors. So this type of sensors exists, and they are widely used in all research labs. They have been a bit late in arriving at the market, since the technology necessary is extremely expensive, because it includes mechanic parts with very high precision. So our approach aims to overcome this problem and try to achieve a similar level of resolution using low-cost technology. The second case, regarding pregnancy monitoring, uses electrodes to perform a non-invasive analysis of the uterus to extract electric signals that will then be analyzed to determine the health states of the woman and the baby. So the first prototype is based on metal nanoparticles. The system is extremely simple, as we can see on these pictures. There is a source of light, a bulb, which transmits light through a plasmodic, and then there is a camera, which then takes a photo. The photo is then sent to a processing center where there is a color analysis, and any color change has to do with the amount of biomarkers, which are practically immobilized by antibodies. The approach, using the other approach, is a little more sophisticated, because it involves manufacturing technology, more sophisticated, more complex. But once again, the system, the extraction system, is extremely simple and has to do with a comparison of the intensity of light, which is issued to different channels. So the signals are acquired through a sensor, go through a computer, and then sent to a remote system. The Uterine Explorer, this is the prototype of the software, which is in charge of this electric signal to monitor a pregnancy. And this is an algorithm that was made based on real data in Iceland, and also in Lisbon at the Alfredo da Costa maternity. In terms of conclusions, our point of view is that these technologies are being used in mobile phones and social media, but they might also be used to help people who live in remote locations with scarce access to healthcare, and they might also be used to alleviate the workload of hospitals, because the same technology might be used for a high monitoring of the patient at home without the need of the patient having to go to the hospital physically. Obviously, these are our results. They might be improved. Our plan is to carry out and carry on working and include other cases, other types of diseases, so we can analyze them, and we will also try to be multidisciplinary, so we can include different materials with photonics technology and different types of technology. Obviously, the IT systems for the transmission of data. Thank you.
Obrigada. Eu queria agradecer a todos os oradores pelas.
I would like to thank all speakers for their excellent presentations, and also to speak within the allocated time. I would also like to say that it's very interesting in a public health session that the topics have been immunology, in artificial intelligence, and so on, which shows how we've been evolving in public health and how the future of public health will be for the health of populations.
Podíamos agora ter aqui momento.
We could have now a moment for some questions. We need to prepare the room for the closing session, but if you have any questions, please feel free.
Sim, podemos. E é interessante. E tem aqui, se faz favor, o microfone. Pode falar que liga.
Já está ligado. Eu tinha 3 perguntas, posso dizer todas? É para aqui.
I have some questions. I have a question. An attitude we have in medicine, trying to be affectionate toward people. From what I understood, having attachment. This is for Ricardo. So with our colleague who works with pets. So it means that our pets might transmit bacteria. I didn't quite understand. Wasn't not the other way around? But this person that you have at home did not develop at home. How is this then pursued at the hospital? And then the pregnancy. Where do you place the sensor on the pregnant woman's womb, and what exactly is it measuring, so I can better understand it? Thank you.
Thank you for the question. The connection in psychology is a concept that can be considered in other areas. Children are born with a certain behavior to guarantee survival. And this doesn't happen only during the first years. This attachment is then conveyed to other figures of... it replicates in the loving affairs and relationships with others. We have to consider that they use this attachment in adult age. People who feel more anxious in terms of attachment with others, they tend to involve with more COVID risk, because these people, because they don't have an autonomy regarding the primary attachment figures, the fact that people are not there present they will not give them affection and love. They always need this positive love from others who are there to give them comfort, love, and tenderness. But on the other hand, the fact they are in a lockdown and have not the opportunity to socialize with others created more anxiety. That's why some people had the need to socialize with others because of this anxiety. I don't know if I was able to reply to your question. It's the opposite. The result for us was very curious. People who are more impulsive would be involved less in risk behaviors. This shakes some structures, some ideas that our psychologists think impulsiveness is always a risk. Sometimes more recent models say that some risk factors with former theoretical models we thought that people had more predisposition for risk. Also, the context, if the context is different than the pandemic context was really different and new, if we can generalize this result, the fact of someone being impulsive, it doesn't mean that the person is going to be involved more in risky situations. Even the opposite happens. Thank you for your question. Transmission and flow of transmission of the resistance genes have to do with the resistance mechanism and to what type of antibiotics, for instance, cholestyna. Does it depend on the resistance to the type of antibiotics? In the case of cholestyna, what we have observed is that the percentage of the resistance to cholestyna in these dogs and cats that socialize with their owners on a daily basis, the level of resistance is higher than the resistance we find in people in the community. The study at the consortium, we did several cohorts. People are healthy. This transmission was in healthy people. These genes exist in cats and dogs, in the intestine, in bacteria. We isolated the resistance, which can or not be pathogenic. In this case, had several viruses leading to the infection of the urinary tract. Well, I would like to be able to reply to all those questions, really, through the environment, through the atmosphere. We don't really know the passage by physical contact, surfaces. We are speaking about plasmodiums that have a resistance to cholesterol. In the case of people, if they have this resistance mechanism in their intestines, being ill or being sick, or if they do surgery, possibly they can go to places where there are more bacteria, producers of KPC that are endemic in the Portuguese hospitals. In practical terms, it's very simple. Transmission, and we are speaking about transmission, doing genomic studies regarding the bacteria, and the bacteria has variations, polymorphisms, which are very tiny. For instance, we are speaking about E. coli that had a plasmodium, one or two of difference. The bacteria are alike, are very similar. They do measurements of hygiene, like in hospital, washing hands. I can elaborate a little bit more. In this project, what did we realize? Between healthy animals and healthy people, the transmission was very low. Then, what did we realize? Cats and dogs with infections in the urinary tract, the pathology bacteria wouldn't go from animals to mankind, but selecting the treatment. Many multi-resistant bacteria would be in the animal, and people had the same resistance mechanisms. Therefore, the selection of a use in a certain division, when animals are sick with infections of the urinary tract and skin infections, it was bigger when there was a selection pressure by the use of antibiotics, even bigger if there were, according to the type of infections. In case of skin infections, colonization, a sick animal with Staphylococcus with resistance with a bacterial resistant to third generation condition, this possible transmission. If it was easy, we wouldn't be studying the subject, but there is no danger to have animals or pets. When they are taking antibiotics, we have to control infection with physical barriers and hygiene when we deal with these animals. Thank you.
É só falar? Pronto, sim. Pronto, a sua pergunta é o que é que isso vai fazer com a mulher grávida?
So let's now see what we can see with what we can do with a pregnant woman. So the objective is to place some electrodes on the pregnant woman's belly. It has to do with the uterine contractions for a period of half an hour. It will collect these data and then it will be worked by the algorithm, which will assess then the health state. The algorithm might be anywhere. So this woman who is in a village, she will send it to a remote signal and then she will have access to this type of information and then it will be sent to her mobile.
Acho que podíamos continuar aqui a tarde toda, mas vamos ter que terminar.
We would be able to spend here the whole afternoon, but I think we need to end soon because of the closing session. Thank you very much.